receive my presentation?
Okay. Yeah, we can see the presentation.
Okay, thank you so much. Thank you so much to the organizers for this wonderful opportunity to be here today and be able to share some of our research on bacterial endosymbionts of arbuscular mycorrhizal fungi. So like Rafa said, I am an assistant professor in University College Dublin. And so I'll give you a little bit of background on where it is that I work. So University College Dublin, for those of you that don't know, it is located just outside, I'm actually going to get a pointer, just outside of Dublin City on the east coast of the island of Ireland. And it is Ireland's largest university with just under 40,000 students. Just under 12,000 of those are graduate students and around 2,000 PhD students. So the School of Biology and Environmental Science, where that I'm part of, is housed in this wonderful new building here. It is called the O'Brien Centre for Science. And it actually is a complex, so it's a complex of 4 buildings, and they have these wings called the North, South, East, and West Wing. This is the West Wing of the O'Brien Science Centre, which is home to biological research. This is where my lab group is located. It's also home to many other very cool biological research groups, and also has some wonderful teaching spaces as well. So the main theme of my research is inter-kingdom interactions. I'm particularly interested in how bacteria and fungi interact with one another as part of the plant microbiome. So plants, of course, we know are populated by diverse communities of microorganisms, including bacteria and fungi. And so I'm interested in how these bacteria and fungi interact with one another and how these interactions affect their own individual biology, as well as that of their plant host. And my research attempts to understand these interactions at different scales. So I'm interested in studying them at the scale of molecular biology, but also at the scale of ecology. And so whereas I'm interested in all types of bacterial fungal interactions, my main research focus is on bacterial endosymbionts of fungi, also known as Endobacteria. So Endobacteria, the term really refers to any bacteria that lives inside the cells of another organism, in this case, a fungus. Oops, what happened? Somehow my screen became small, apologies. What did I do?
Maybe
I'll reshare my screen, apologies. There, sorry about that. I tried to click something that said hide it, but instead it just decided to go away completely. Okay, so Endobacteria, fungal Endobacteria, they live inside, of course, fungal cells. And so here's an example of what that looks like. This is a microscopy image of a fungal hypha that is packed full of these bacterial endosymbionts. Here they're appearing in green. Fungal endobacteria can also live inside fungal spores, and that is how they're vertically transmitted from one host generation to the next. And you can see here 2 fungal spores which are also densely packed with these endobacteria. And besides being, super cool and interesting to study from a point of view in symbiosis, fungal endobacteria or endobacteria are actually quite common in fungi, and perhaps even more common than we had originally thought when I started my work on bacterial fungal symbioses back in 2011. So there's a study that was published in 2021 by Robinson and colleagues who did this large-scale screen of hundreds of fungal isolates for presence of any type of associated bacteria. And these fungi were derived from either culture collections or genome sequencing projects. And of all of them, it turned out that 88% of all of these fungal isolates were actually associated with some kind of bacteria. And these fungal isolates belonged to all major fungal phyla, including the Ascomycota, the Basidiomycota, the Mucormycota, and all the early-diverging fungal groups as well. And then the authors did some microscopy confirmation to show that actually a lot of these bacteria, or many of them, were in fact endohyphal, so they were endosymbionts, kind of leading us to kind of changing our worldview a little bit in that apparently bacteria associated with fungi are actually the rule rather than the exception. And so despite the fact that they're common and ubiquitous, our knowledge of these bacterial fungal symbioses does remain limited, and this is where my research interests come in. However, even though we don't know a lot about it, what we do know is that endobacteria can actually play profound roles in fungal biology. For example, endobacteria can affect the reproduction of their host. In the case of this fungus called Rhizopus microspores, if you remove its bacterial endosymbiont, it loses its ability to reproduce sexually and for the most part, asexually, and for the most part sexually as well. Endobacteria can also affect fungal metabolism. For example, it's been shown that endobacteria can affect fungal lipid metabolism and reactive oxygen species metabolism. Endobacteria can also play a role in defense by producing the special toxins that will act to protect their fungal host from their own predators, which are typically things like nematodes and amoebae in the soil. And there is also tantalizing evidence that endobacteria might affect the interaction between their fungal host and plants. And so my current research interest is in arbuscular mycorrhizal fungi that Andrea so wonderfully introduced. So I'll just reiterate with a little bit of background about this group, and that arbuscular mycorrhizal fungi, AMF, they are ancient plant root symbionts, all belonging to this Glomeromycotina subphylum. They are nutritional, mainly symbionts, providing their plant host with essential mineral nutrients in exchange for up to 20% of photosynthesis-derived carbon. AMF are ubiquitous and they're very, very widespread, and it's estimated that plants with AMF actually make up around 70% of the global plant biomass, kind of arguably making it one of the most important symbiotic relationships on our planet, with these overarching impacts in terrestrial ecosystem functioning and sustainable agriculture as alternatives to this non-renewable and energy intensive to produce mineral fertilizer. AMF also have endobacteria, and to date, 2 different types of endobacteria have been described in AMF and have been named as these new species of bacteria. So the first one is called Candidatus glomerumbacter gigaspororum, or Glomeribacter for short, as I'll refer to it today. Glomeribacter is the better studied endosymbiont of AMF, and it appears to possess these characteristics of mutualists, and that Glomeribacter appears to prime the energy metabolism of AMF. It can increase the hyphal proliferation during spore germination, as well as the sporulation success of their AMF host. The other endosymbiont, called Candidatus moeniplasma glomeromycotorum, or moeniplasma for short, is more of an enigma, in that there's still no experimental characterization of what their role actually is in AMF biology. In other words, we have absolutely no idea what moeniplasma do in AMF, but they do appear to possess some characteristics of parasites, specifically their molecular evolution patterns, and their population structure is indicative of maybe their parasitic nature. So when I began my work on AMF and endobacteria, everything we really knew about this AMF relationship between AMF and endobacteria came from studies of AMF, which were themselves derived from culture collections. And nothing was really known about these AMF-endobacteria relationship in nature. So I wanted to know how common are actually endobacteria in nature, and what are their distribution patterns of these endobacteria in natural AMF populations. So to answer these questions, I went out and I did some soil sampling at different sites, some of them in Ireland, some were in the States. And from these soil samples, I extracted AMF spores, and then I screened them for the presence of the 2 known endobacteria, the Glomeribacter and the moeniplasma. And what I found was quite interesting. I found that Glomeribacter, the one that is a mutualist, was actually very, very rare in these natural AMF isolates, found in only about 1% of all of these spores. Moeniplasma, on the other hand, the one that we don't actually know what it does, maybe it's parasitic, was found in 80% of all the isolates. And this pattern, this is not an average from all of these sampling sites. Some of them were separated by thousands of miles. They had different soil chemistry, different plant community structure, and they even had different underlying AMF community structure. But this pattern of Glomeribacter being very rare, moeniplasma being very common, was the same. So this pattern must indicate something, and it's likely indicative of the respective roles of these endosymbionts in AMF in nature. The other thing I wanted to highlight is that because myonoplasma is so common in natural AMF populations, it's very unlikely that they are actually truly parasitic. And in fact, my personal hypothesis is that they are not parasitic, that they're likely have some kind of a benefit for AMF. We just don't know what it is. And so this is one of the major directions of my research group, is trying to find out what myonoplasma actually do in AMF. Now, you might be wondering how we actually do these kind of distribution surveys of Endobacterium. So I thought I'd give you an example of our typical spore processing workflow that we use in the lab. So we start out with a collection of AMF spores, typically isolated from the soil. And then we have to clean them. They undergo a surface sterilization protocol because we're interested in Endobacteria, so the ones that are found inside the spore. So we not want to be characterizing anything that's attached to the spore in some way. We then, after the surface sterilization, the spores get manually picked and placed in individual PCR tubes with 1 spore per tube, and they get manually crushed using a pipette tip to release the spore contents, including the DNA. And then they undergo a whole genome amplification process because the amount of DNA in a single spore is really not enough to do any meaningful downstream molecular work. So after whole genome amplification, we do typically PCR with specific primers. So for example, if we're interested in a particular AMF spore harbors myonoplasma, we can do PCR with myonoplasma specific primers. If we're interested to see if a particular AMF spore has any type of bacteria, we can do PCR with universal bacterial primers. And so then we can send that product, the PCR product, off for sequencing to gain some understanding and information about what actually are the population structures of these Endobacteria, or even the community structure of these Endobacteria like. So this process is quite laborious, but it's quite powerful because it gives us spore level resolution of these bacterial populations or communities, and we can link them to the individual AMF genotypes. So using the spore processing workflow, we decided to characterize the entire diversity of Endobacteria associated with these AMF spores from nature. And so we took our spores that tested positive for any type of Endobacteria in our PCR-based screen, and we subjected them to bacterial community profiling using this 16S amplicon sequencing on the Illumina MySeq platform. And what we found was very unprecedented. We found that these AMF spores were actually home to whole communities, to very diverse communities of Endobacteria. So what you're looking at here are bar plots showing the relative abundance of bacterial phyla inside these AMF spores. And on the left is a kind of an extended bar plot with each row corresponding to a single spore sample. So you can first of all see that these plots appear red. And that is because in red are bacteria belonging to this Tenerocutis phylum. It's now called the Mycoplasmatota phylum. These are our myonoplasma Endobacteria. So you can clearly see that myonoplasma Endobacteria are the most commonly found inside these AMF spores, and they also have the highest relative abundance inside individual spores. The Glomeribacter Endobacteria, they belong to this Proteobacteria phyla. It's now been renamed to Pseudomonodota phylum. But the Proteobacteria you actually see on these plots are not Glomeribacter. Glomeribacter was so rare and so uncommon that you actually can't see them on these plots. I really had to work to find them in the data set. But besides that, you can see that these AMF spores are actually home to bacteria belonging to 10 other phyla. And in fact, we found that a single AMF spore could harbor up to 277 other bacterial quote-unquote species or OTUs that were not Glomeribacter or myonoplasma, not the already known Endobacteria, with an average of about 25 of these bacterial taxa or species or OTUs per spore. And what were they? Well, they were actually belonged to known free-living soil or aquatic bacteria, and were actually previously unknown to have any type of endofungal lifestyle. So this sort of changes our view of AMF as being hosts to at most 2 different endobacteria. And it turns out that in fact, AMF can be hosts, or at least AMF spores can be hosts to whole communities of endobacteria. As to what they're actually doing in AMF, we also don't know, but we have plans to find out. So then we looked at the populations of the mycoplasma endobacteria in a little bit more detail, because these are the ones that are more prevalent and more of an enigma. Now, mycoplasma are quite unusual, and this has been done for quite some time, that mycoplasma populations can be very, very diverse, even individual AMF spores, with at most 3 different kind of mycoplasma genotypes, or OTUs, being known to coexist in a single AMF spore. Glomeribacter, on the other hand, the other endosymbiont, their populations are always genetically homogeneous inside individual AMF isolates. Well, using our this bacterial profile, the 16S profiling data, we were able to show that AMF spores could actually be home to as many as 50 different mycoplasma OTUs, or genotypes or species, with an average of 22 mycoplasma OTUs being found per spore. Kind of really highlighting the unprecedented diversity of even these endosymbionts in natural AMF spores, not natural AMF spores, AMF spores from nature. And so analyzing the mycoplasma populations in a little bit more detail, we noticed an interesting pattern. We found that the mycoplasma population structure, their richness, their incidence rates, and their intraspore relative abundance were all related to the AMF host genotype in some way, leading us to conclude that AMF host genotype must be a significant driver of mycoplasma population assembly into these spores. And this is analogous to what has previously been shown in other eukaryotic systems, where host genotype is a known significant driver of microbiome community structure in animals, including humans, in plants, and even in fungal fruiting bodies. And it turns out that this also now extends to AMF and their endobacteria. So with that, I was going to spend a couple of minutes telling you a little bit about the main directions of my research group at the moment. So like Rafa said, my research group is called the Microbial Interactions Group, or the MINT Lab. It is just over 2 years old now. And the main project in the lab at the moment is really trying to functionally characterize these mycoplasma endobacteria and try to get at their role in AMF biology and their role in the AMF plant interaction. So this is a project that's funded by Research Ireland, and the idea is that we will obtain these comparable isolates of AMF with and without mycoplasma, and then compare, the growth, their patterns, basically their biology and their interaction with their plant host to glean some kind of an idea of the function of mycoplasma in AMF. The other main research direction is on the ecology of the AMF endobacteria. And we want to, among other questions, there's just a few questions that we would like to answer. For example, what are the environmental drivers of mycoplasma distribution in AMF? So in the past, I've shown that the probability of harboring mycoplasma in certain AMF taxa actually increases with increasing plant density. And we recently have some preliminary data, which I didn't want to show yet because it's very preliminary, but it appears that mycoplasma might also, their prevalence might, or their likelihood of finding mycoplasma in these spores will actually decline under drought. Another question I want to address is how are actually endobacteria assembled into these AMF spores? Where did they come from? Did they come from soil? Did they come from the AMF hyphosphere, or maybe the root endosphere? And also, what are their ultrastructural dynamics inside AMF? So when an AMF spore that's full of these endobacteria germinates, where did the endobacteria go? Or how did they actually get assembled into new spores? Where did they come from? And so to answer these questions, we have some really nice research infrastructure at UCD. So we have these great state-of-the-art molecular environmental labs where we do all of our spore processing and all the downstream molecular analysis. We also use these facilities for in vitro cultivation of our AMF isolates using root organ cultures. And we've also started to play around with asymbiotic cultures as well. As part of the UCD main campus, we also have the UCD Rosemount Environmental Research Station, which includes these state-of-the-art glass houses where we, for example, we will grow up plants, specifically where we do trap cultures for AMF, but also where we do any plant-based experiments. And then, not very far away from the UCD main campus, is this UCD Lions Farm. And UCD Lions Farm is very cool because it is an active farm, but it's also a research farm. It's a hub for many different field-based projects that UCD faculty carry out. And with that, I am going to finish. Before I finish, I do want to give a shout out to my funding sources, Research Ireland, the UCD School of Biology and Environmental Science, and also the China Scholarship Council, which funds 2 of my PhD students. I want to really thank my wonderful collaborators and mentors over the last few years who have helped me in setting up my research group and my current group, which has 3 PhD students, Yueze and Elise, and Sean, who is a research assistant, and also all the various interns and visiting students and undergraduates that have come through the lab over the last 2 years and contributed really nicely to the group dynamic and the research. And with that, thank you all for your attention. I'd be happy to take any questions.
Thank you, Olga, also for a very nice presentation with a lot of novel and revolutionary insights as well. It's very nice to see all the bacteria and hyphae. I had no idea that they were so abundantly present. So yeah, if there are any questions, feel free to ask in the chat, or as I can see, Nabla is here already. Go ahead.
Hi, Olga. It was really interesting. Thanks for the presentation. I'm just sorry if I missed it. Can the mycoplasma live independently, or are they obligate symbionts of the fungi?
A good question. They do appear to be obligate symbionts, that they're uncultivable. We haven't been able to culture them in the lab, and there is no indication that they exist outside of their fungal host anywhere.
Okay, cool. Thank you.
Yeah. Question.
I also see that Andrea asked a question in the chat with regards to the molecular evidence you have of the bacteria inside of the spores. Do you also plan to supplement it with morphological evidence, or do you have that already?
No, that's a great question. That's something that we that is not actually part of the current project, but the next project that I will get funding for is to actually do fish and try to understand where they are in the spores and what they do when the spore germinates. Yeah, and we want to do that for both mycoplasma and a lot of the other ones. So for example, we could get fish probe designed for targeting different bacterial phyla. So to see kind of how they're distributed. Are they all together in the spore, or are they somewhere not together? Yeah, that is definitely really cool. It is quite challenging because the spores, I don't have expertise in this, and I've talked to some people who are experts in this, and we hope to collaborate in the future. But apparently the spores themselves, because they have such thick cell walls, it's very hard to get a probe to penetrate inside the spore. So that's a challenge that we'd have to address with our experiments.
Yeah, sounds like very interesting experiments are indeed ahead. I can also see a question by Didier in the chat as well. So can these fungi be cured from their endobacteria, and how can you exclude surface contamination?
Yeah, that's a very good question. I'm just gonna...
Oh yeah, I see indeed Didier joined us as well.
Oh, hi. So can they be cured from the endobacteria? That is something that we are hoping to achieve with this project. And we have some ideas of how to achieve it, but it is a quite challenging system. If AMF are obligate biotrophs. They need a plant host in order to be cultivated in the lab. And so And their growth cycle is quite slow. So yeah, this is something that we're working on at the moment. I know that from not my PhD work, but when I was a PhD student, there was a student, another PhD student in the lab, Mizue Naito, and she was trying to cure mycoplasma from AMF using antibiotics. But I know that, I've seen her struggles. It was unsuccessful. So we're going to try a different strategy.
So what about using those that don't have it, the rare cases that, and the basically most isogenic relative fungi that have it?
Yeah, that's one of our we have a couple of approaches that'll be one of our approaches. One of my PhD students we currently have a collection of Rhizophagus claris isolates in the lab, and some of them have mycoplasma and some of them don't. And we kind of want to, besides actually sequencing their genomes, which is challenging at the moment and a whole other challenge, we do want to get some kind of a phylogenetic reconstruction of the relatedness among these Rhizophagus claris isolates, and then from them try to pick ones that do and don't have endobacteria, then hopefully they'll be comparable enough. Yeah.
Great, super. Very exciting. Thank you.
Thanks.
Okay, I guess that also answers the other question we have in the chat of how you proceed without affecting the spores during the surface sterilization process.
Yes, sorry, go on.
No, go ahead.
We do have, so the surface sterilization protocol that we use was developed to keep the spores alive. So it is a gentle enough process that it doesn't kill the spores, but it also doesn't rupture them. Although some of them do rupture, and then we just end up not using those. Yeah. The protocol is published, so whoever's interested, I'm happy to share it with them. You can email me.
See that someone is typing. In the meantime, I also had a question concerning the entire microbiomes essentially that you identified. Did you analyze whether there are also any propensities for certain biochemical processes that are overrepresented in there, as to get an idea of what they could do? Or
no, that's a great question. I think that would be super cool to do, but we use 16S profiling, so we only know who it is. We didn't do metagenomics, so we weren't able to analyze the other genes. But that's a good question. I would imagine it's something to do with lipid metabolism, because they're probably feeding on the wonderful, delicious lipids that are in there.
Yeah, that will be very interesting to assess.
Yeah.
All very exciting. See, Rafa joined as well. Is it with a question, Rafa?
No, I already had, I had the same Nabla, so it's okay. I already done one, so yes.
I still see some people typing in the chat as well. I see it's all very interesting, of course. Yeah, also I think there's always a possibility also of contacting Olga as well, should any more questions arise. Yeah. I see another question. So do these endobacteria have a direct effect on plant growth?
That will be wonderful to find out, and something this is part of the main projects that we're working on. Yeah, we don't know.
Okay, great. A lot of interesting questions again lie ahead, so that's nice. So I guess that concludes it then for today. Yes.
Yeah, I think so. Go ahead, Nami.
Okay. I was thinking maybe you could tell us the other meetings that are still coming. So the next round, of course, we have a very nice schedule already ready for the speakers to come in the coming few months. And I think in November the 28th, there's already the next one coming up. So it's quite soon now, also to avoid the holidays a bit. And then we will have two speakers again. So Dr. Victor Carrion from the University of Malaga, as well as Dr. Margarida Santana from the University of Lisbon. But of course, more information of that is still to come. As always, you will see us appear in your mailboxes as well. So then finally, I'd also, of course, like to thank our two speakers of today again, Andrea and Olga, for both very, very nice presentations. And everybody for being here.